Thank you very much, Anna, and obviously Deborah, but we can't see Deborah at the moment. So that's the first of 3 talks then given. So now I would just open the floor for any questions anyone has. Perhaps you can put them in the chat. I can't see anything at the moment in the chat about any questions. That means that everybody's all very clear on what we spoke about. Any comments or anything? Deborah did say, because she's not here, but she is happy to receive any questions by email. We can forward them to her if anyone has something to ask about her talk as well. So I can't see anything at the moment. We're all on time as well, I believe. The break is in 5 minutes, so you've got 5 minutes for any questions. I guess if there's no talk, no questions. Oh, here we go. Oh, Andreas sent a message. You can also unmute yourself if you have a question. Perhaps everybody's being shy. Oh, so a question from Bill. Do you focus on proteins that were antigens based on immune responses? Is this for Anastasia, Bill? I presume based on the OMVs. Do you want?
Yes, I'm sorry. Yes, it is.
So I chose my list of OMV proteins based on, there was a, Dio et al. Published a paper where he had extracted proteins from gonococcal OMVs, and he had a list of 110, but because I was relatively limited in my time scale, I selected an initial list of 25, kind of prioritizing outer membrane proteins because they're more likely to be involved in AMR, or as you say, immune responses. And then it was kind of based on abundance and things like that.
Great, thank you.
The creation of these loci is really quite time consuming. I just want to put that out for all the curators who work on this, and Anna has been curating a lot of these genes, and Anna, who we're talking this afternoon, Anna Sehoven, has been curating some other genes, and it's actually quite a time consuming process. So you do, for Anna's project, we did have to kind of limit the amount of genes that she was looking at, because otherwise you'd be curating for years. Really wouldn't.
It's quite odd because a lot of the genes will be relatively easy because there's so much annotation that's already been done, but there's always the more difficult ones that throw up problems. Like we had a particular gene, I think it was nice 1428, that had different start codons. There were different alternative start codons, so that caused some problems with curation. So that took a lot more time than other genes that were much more readily curatable.
Yeah.
Oh, you're muted, Martin.
Yeah, I am, aren't I? We got a good we've got a good comment here from Yolinda, who we'll be hearing later about. Are you aiming to identify vaccine targets? I mean, the interesting, of course, the interesting point, and one of the advantages with PubMLST that we use genus schemes. So obviously meningococcus and gonococcus exist in the same database. And of course, many people know that there's a lot of interest, and Charlene can't be with us today because she's on maternity leave, which is a wonderful reason for her not to be with us. But obviously there is interest in meningococcal vaccines being used against gonococci. So Anastasia, yeah, would you like to say a bit about the antigen genes and vaccines?
Yeah. Now that's something I would have really liked to look into, but as I'm funded by the BBSRC, I had to focus on a more AMR directed angle, but I definitely think there's potential for the scheme I made or further schemes to look at vaccine targets and things like that. That's, yeah, as Martin said, that's something that Charlene was kind of already beginning to look at with her scheme. But yeah, unfortunately, because I'm BBSRC funded, you have to move away from kind of directly medical things. So that's why I was looking at more antibiotic resistance association with OMVs.
Yes, just to explain to overseas visitors, yes, the BBSRC in the UK is the Biological Research Council, and it allegedly doesn't support medical research, so hence they are interested in AMR. So, but there is a lot of interest with Charlene did run up a meningococcal outer membrane vesicle scheme, and of course a lot of these antigens are common, and there's increasing evidence really that the Bexsero vaccine has some impact on gonococcal disease, and that must be through these through immune response to these proteins.
Sylvia Brewston for Anastasia. The deduced peptide sequences do not show tertiary structures. Is there a possibility to also look at this by known folding schemes?
There's definitely a possibility. It's something you can do as a kind of further on step, as you can begin to look. There's data out there on how these proteins would fold up and what their structure might look like. Unfortunately, it wasn't within the reach of my project, but it's definitely something you could do further along to kind of get a little bit closer to actual phenotype and to how these proteins might actually work, or the significance of those changes. Like if you see a particular variant has really taken off in a particular country of a protein, it'd be really cool to see if there is a structural reason why it's been more beneficial for AMR or other reasons.
There's an interesting comment from Paola. Hello, Paola. Good to see you. Maybe coupling a comprehensive list of vaccine candidates being examined could help to focus efforts. Well, indeed, we can do this. And one of the advantages of the MLST scheme, the PubMLST platform and the way it's run, as Odile was describing earlier, is we can have any number of schemes within the database which contain any number of loci, which can be either peptide loci or indeed sequence loci, and because that's basically just a text list, we can have as many of those as we like, and quite a few of them exist, don't they, Odile? But
I think a lot of them would have been defined already. I guess there's more complicated, like Anna was saying, the OPA proteins. They're a bit more complicated to work on in draft genome data. So there's some loci that will be always a bit more tricky or challenging, let's say, to look at. But certainly I think as a kind of general sort of consortium, we could put together a list of antigens that people are interested in and create a scheme to look at those.
And if anybody's interested in having a scheme and curating it, they're very easy to create, and we're very happy to accommodate that. And that would mean that all of your proteins of interest or genes of interest would be in that scheme, and you could access it very easily within the database. So, yeah, we'd be very happy to do that, Paola.
Okay, so we're into our tea break or coffee break time. So I guess we'll have there's no other questions in the chat for anyone. So yes, I think we'll just have a short pause and meet back
it's
here.
5 minutes at
What time were we meeting back at? It was 3.
10 minutes.
Long enough pause for everybody?
At 1500.
Yes, we restart at 1500. So all go and make yourselves a cup of tea. And we'll all see you very soon. And there's over 50 people. Wonderful to see you all. Thank you for joining us, and see you again very, very shortly.